Hello, thanks for tuning in my talk. My name is Rich Price. I'm a professor of biomedical engineering at the University of Virginia. And the title of my talk is Immune Response to Blood-Brain, Blood-Tumor Barrier Opening with Focused Ultrasound. So my presentation is going to have two parts. In the first part, we'll talk about some work we've done sort of profiling the immune landscape after blood-brain and blood-tumor barrier opening in models of both melanoma metastasis and a primary tumor model, glioma. And then in the last part, I'll have a few slides on some recent work we've done using blood-brain barrier opening to deliver an immunotherapy CD47 across the barrier to control glioma. So for this first study, I'm going to be very brief. We published a paper last year in Theranostics, so I'm only going to show a couple of slides on it because the interested viewer can always go check the paper out. But in essence, we open the blood-brain and blood-tumor barriers in and around B16, F1C, ova, and other melanoma tumor models implanted in mice. And again, we open the barrier using the parameters shown here, which are pretty standard in the field. We then examine the tissue using bulk RNA sequencing at both 6 and 24 hours. And then we follow this up with flow cytometry to look at different immune cell populations at both 2 and 4 days. So this is basically a study of a relatively acute response to blood-brain and blood-tumor barrier opening. And the flow cytometry here was essentially done in the meningeal tissues, tumor, as well as the draining lymph nodes. So in that, again, published paper, we came to a few conclusions. So the first was that, when we open the barrier with focused ultrasound, we do see a significant enhancement of pro-inflammatory transcripts as well as gene sets associated with immunity, consistent with the sterile inflammation response that others have observed before. When we used flow cytometry, we noticed that there was a very, very modest but still significant enhancement of dendritic cell maturation in the meninges. There were signatures of enhanced dendritic cell antigen processing in both lymph nodes and the tumor as well. But again, it was pretty mild. And none of this really translated into a T cell response. So there was no activated T cell homing. And there was also really no change in endothelial cell adhesion molecules. So again, it was a essentially modest but transient response after barrier opening in the melanoma metastasis model. So another study that's now in review is essentially shown here. I'll go into this one in a little bit more detail. Very similar to the previous study, we opened the blood-brain and blood-tumor barriers, except this time we're using a primary tumor model, GL261 glioma model. The parameters are the same as before, except that we're using slightly different pressures. We're using a slightly lower pressure of 0.4 and a slightly higher pressure of 0.6 megapascals. And we're looking a little bit later and only using flow cytometry. So now looking at day 7 and 14, again, doing flow cytometry for meningeal tissues, the tumor, and the draining lymph nodes. So the first thing we noticed is that there is, again, some modulation of dendritic cell maturity as well as representation. So representation basically meaning the number of percent of cells that are in the given tissue. So when we look at the number of dendritic cells in the GL261 tumor, there is a trend towards a significant increase at the higher pressure of 0.6 megapascals. In the draining cervical lymph node, there actually is a statistically significant increase of 0.4 megapascals. And interestingly, we actually see a drop-off in dendritic cell numbers in the meninges, potentially, and this is a speculative statement, so take it with a grain of salt, that dendritic cells are actually trafficking from the meninges down into the lymph nodes and or even back into the tumor microenvironment. When we look at CD86 as a marker of dendritic cell maturity, we do see an enhanced signature of maturity in the tumor at the higher pressure, but no other signs of changes in maturity in the other compartments. However, much like we saw with the melanoma model, there's really no translation of these dendritic cell responses into changes in T cell representation. So now looking at both days 7 and 14 in both the GL261 tumor as well as the draining lymph nodes, we really see across the board no change in CD8 or CD4 T cells with the exception of a slight but still significant dip at 0.4 megapascals. And in concert with this, there were really no evident changes in T cell function. So if we stain for granzyme B and or interferon gamma, we again see really no significant changes or patterns in the T cells. We do see something going on at least mildly when we look at checkpoint inhibitors. So we specifically looked at PD-1 and TIM-3. So I think most people are familiar with PD-1, which can engage PD-L1, which can be expressed by tumor cells and other myeloid-derived suppressor cells or other suppressive cells in the microenvironment to essentially reduce T cell function. TIM-3 is similar when it binds to collected 9, which can be expressed by myeloid cells or indoor tumor cells. It can essentially lead to T cell exhaustion or anergy. With that said, they're also essentially both markers of activation. So, the molecules are now expressed when the cell becomes active, which essentially now puts in place a mechanism to turn the cells off. So when we look at PD-1 positive cells, this is at day 7, we see a trend towards an increase in PD-1 expression at the higher pressure. There actually is a significant increase in PD-1 expression with the in the 0.4 megapascal group. And interestingly, that actually translates to the TIM-3 group as well. So at this lower pressure, we're actually seeing, again, sort of an enhancement of both PD-1 and TIM-3 expression on the CD8 T cells within the tumor. But by day 14, it is essentially quiesced again. We're not seeing any differences. So in conclusion, when we open the blood-brain and blood tumor barriers with a couple of different negative pressures in the GL261 model, we see changes in the number of dendritic cells in the tumor, draining lymph nodes and meninges. At different pressures, it seems to go up a little bit in the tumor and increase a little bit in the draining lymph nodes, and it actually goes down in the meninges, again, potentially suggesting that there is trafficking of the dendritic cells from the meninges either to the tumor and or to the draining lymph nodes. The maturity of the dendritic cells does improve in the tumor, but again, we see no change in T cell representation or function. Yet there is signs of activation here, albeit moderate. So there are significant, but again, slight changes in the expression of PD-1 and TIM-3 on CD8 T cells, and especially at the lower pressure here. Okay, so I'll move on to the last part of my talk now. So I'm going to change gears a little bit, step out of sort of the immune profiling of the response and look at using blood-brain and blood tumor barrier opening as a way to deliver an immunotherapy. So the immunotherapy that we're delivering here is anti-CD47. So CD47 is a molecule that's expressed very, very highly on most tumor cell types, including GL261 glioma, which is the model we're going to use. It binds to a ligand on phagocytic cells like macrophages and dendritic cells called SIRT-alpha. And when it does this, it essentially sends a dopamine signal. So it is essentially a way for the tumor to control phagocytosis by various antigen-presenting cells. And because this is in place, or let me put it this way, if we were able to sever that connection and block CD47, it would allow a couple of things to happen. First of all, the macrophages and other phagocytic cells could actually potentially control the tumor by through phagocytosis, but the phagocytic process can also lead to antigen acquisition by professional antigen-presenting cells, such as dendritic cells, which could then theoretically allow a systemic immune response to begin against the tumor, which would be wonderful. So these are just some images of tumors before and after treatment in these experiments. So our protocol is shown here at days 14, 17, and 20. We're opening the barrier and delivering anti-CD47 intravenously. A few minutes after opening the barrier, you can see signs of barrier opening with focused ultrasound here. In some of these mice, those are just a few of them. And when we do this and compare the treatment group, which is the dark blue bar here, anti-CD47 and focused ultrasound versus control, focused ultrasound alone, and just the antibody alone without any ultrasound to open the barrier, we see a significant enhancement in growth control with the treatment, and this actually translates very nicely into a statistically significant survival advantage. So, ultimately, we would like the survival curve to continue out to the right a little bit further, and I would argue that the difference that we're seeing here might appear modest. It's more consistent with modulation or enhancement of phagocytic activity as opposed to driving a systemic immune response against the tumor. It doesn't mean that that's not happening. We haven't tested that yet, but this is more consistent with that kind of response. That said, I do think that the power of focused ultrasound blood-brain barrier opening is pretty evident here because we can compare our results to a previous study using the exact same clone of antibody in the exact same tumor model. So about 4 years ago, the Weissman group at Stanford published this paper in Science Translational Medicine, where, again, using the same anti-CD47 antibody in the same GL261 tumor model, they intraperitoneally injected anti-CD47 daily and were able to generate statistically significant increases in survival, such as shown here and here. So the light blue curve is at 16 milligrams per kilogram. So it's interesting if you take our data and overlay that on top of these data, you see that they're almost identical. So our FUS negative curve lines up perfectly with their control, and our FUS positive curve, survival curve, actually lines up pretty much perfectly with their 16 milligram per kilogram dose. But the difference is that we were able to get this survival advantage using far less antibody. So in the previous studies, the investigators had to use about 20-fold more drug without blood-brain barrier or blood-tumor barrier opening to get essentially the same survival advantage that we had. We only did 3 injections at 8 mg per kg. So again, really showing the power of focused ultrasound here as a tool to enhance immunotherapeutic delivery to the brain blood-tumor barrier. So in conclusion, the immune response to blood-brain and blood-tumor barrier opening is modest and transient in models of primary and metastatic brain tumors. And because of that, we argue that FUS blood-brain barrier, blood-tumor barrier really presents sort of a relatively clean canvas for immunotherapy delivery. We don't see, for example, a lot of signs of adaptive resistance, which would be unfortunate because it would mean that there's essentially, again, an adaptive resistance pushing back against any kind of immunotherapy that we were trying to instill here. And then finally, anti-CD47, we believe represents a promising immunotherapy for gliomas in the context of FUS-mediated delivery. So I am going to finish with a couple of burning questions here. So the first one is, I'd really like to know how we can leverage focused ultrasound blood-brain barrier opening to really be more than just a drug delivery system. And the second question really is sort of like a sub-question of that. Specifically, how can we use focused ultrasound to do things like drive dendritic cell antigen acquisition, the trafficking of dendritic cells to meninges in the lymph node, where they can then better potentially cross-prime T cells. So we know that focused ultrasound has the potential to exert a wide array of bioeffects, and exerting those above and beyond drug delivery could potentially manipulate this pathway that dendritic cells have to take in a positive manner, we believe, to actually, again, drive adaptive systemic responses against the tumor, which is sort of the holy grail here. And sort of along those lines, can we leverage immunogenic cell death, which can be created by things like sonrodynamic therapy? So theoretically, sonrodynamic therapy should be able to elicit immunogenic cell death without depleting T cells, which is something that chemotherapies can do. So we're thinking of combining things like chemotherapies with immunotherapies to use that as essentially a tool to drive a systemic response. But perhaps other approaches like sonrodynamic therapy could actually fill that gap. Okay, so that's all I have. So I'd like to acknowledge the students who did the work here. So Colleen is responsible for the most part for the first study. The second and third studies were primarily done by Natasha. Sorry about that. In collaboration with a postdoc in Tim Bullock's lab, Allie Witter, at least on the second study. And then these are my other faculty collaborators at UVA. So thanks for tuning in, and I look forward to the work done.